Hello everyone. Today, I am pleased to introduce the paper entitled Microbial Keystone Taxa and Metabolic Signatures in Centenarians Regulate Intestinal Homeostasis During Aging. Gut microbial networks and keystone taxa are important for maintaining microbial stability and host homeostasis. However, most previous studies on aging-associated gut microbiota mainly focused on abundance changes, while microbial networks and microbe-host interactions were less explored. Highlights: we identified Clostridium-dominated keystone taxa in centenarians and reveals that Clostridium scindens enhances microbial network stability and produces indole-3 acetic acid, IAA, from tryptophan via amidase and aldehyde dehydrogenase, thereby promoting intestinal homeostasis in aged mice through HR-mediated Claudin-10 signaling. In this study we systematically profiled the gut microbial communities and metabolic landscapes across 3 human cohorts involving healthy individuals with different ages. In the older group, the top 10 ASVs that increased in abundance were represented by Pseudomonas, while the depleted ASVs dominated by members of Faecalibacterium, Bifidobacterium. Compared with older group, enriched ASVs and centenarians included the members of Clostridium and Bifidobacterium. Depleted ASVs in centenarians included the Pseudomonas. We next identified microbial keystone taxa by constructing co-occurrence patterns coupled with microbial networks in the 3 age groups. Notably, Clostridium aldinense and Clostridium scindens were identified as both significantly enriched ASVs and meaningful keystone taxa in centenarians. As microbial reconfiguration and network attribution reshape host metabolome during aging, we then examined metabolic signatures. Compared with older group, centenarians exhibited markedly differential metabolome. Notably, centenarians exhibited higher levels of indole-3 acetic acid, IAA, 3-methyldioxindole, and serotonin than those in feces of older individuals. Targeted quantification showed that centenarians exhibited significantly higher levels of fecal indole metabolites including indole-3 acetamide, indole, tryptamine, IAA, indole-3 acetaldehyde, indole-3 lactate, and indole-3 acrylate in feces than relatively older group. Bacterial species in K00128 ALD and K01426 AMI were also predicted to be enriched in centenarians. The genes encode K00128 ALD and K01426 AMI that catalyze the biosynthesis step for IAA, suggesting a possible correlation between Clostridium scindens and IAA production. Further analysis and experiment found that keystone taxa C. scindens in centenarians produces IAA from its substrates by bacterial ALDH and AMI. In this case, supplementation of Clostridium scindens significantly downregulated the levels of cell senescence DNA damage pro-inflammatory cytokines markers, and restored intestinal barrier-associated genes especially Claudin-10 in colon tissues of aged mice. It is worth noting that Clostridium scindens supplementation markedly enhanced the microbial network connectivity and IAA production of colon tissue. Since IAA may mediate these effects, aged mice were further treated with IAA. IAA supplementation similarly reduced senescence and DNA damage markers and restored key intestinal barrier genes, especially Claudin-10. Given that IAA is an endogenous ligand of AHR, the authors used intestinal epithelial cell-specific AHR knockout mice. In these mice, IAA failed to significantly improve intestinal aging phenotypes, indicating that its protective effect depends on AHR. In vitro experiments using senescent intestinal cells confirmed that IAA alleviated barrier dysfunction and DNA damage in an AHR-dependent manner. Finally, molecular simulations and luciferase reporter assays showed that AHR could bind to the Claudin-10 promoter and enhance its transcription after IAA treatment. This provides mechanistic evidence that IAA activates AHR to promote Claudin-10 expression and protect against aging-related gut barrier dysfunction. Summary: this study identifies Clostridium scindens as a microbial keystone taxon and IAA as a metabolic signature in centenarians. C. scindens produces IAA through AMI and ALDH, and the C. scindens IAA-AHR Claudin-10 axis helps maintain gut microbial stability and intestinal homeostasis during aging.